I think that's, is this the same?
No. You have to take this
Okay.
one. This is only for me. Okay.
Everybody can hear me all right? Yes. Okay. So I've been doing science for over 60 years, and every 10 years I seem to change my field. So I was working on aging for a long time, and then I decided that the low-hanging fruit in disease prevention is in nutrition. So the last 10 years I've been dabbling in nutrition, and I'd like to tell you what we found out. What do you? Okay, great. So this is your metabolism. And so what do you need to run your metabolism? You need fuel and oxygen so you can generate your kilos of ATP every day to power your muscles and your brain. And then you need about 40 micronutrients. These 15 minerals or so, 15 vitamins or so, some essential amino acids. And if you don't get any one of these, you die because some protein needs it for doing an essential function. And what we know is how much we need to keep from dying. What we don't know is how much we need for a long, healthy life. And I'm going to tell you what I think is the principle of this and why the whole world practically is not optimum in this. Okay, so these are let's see. I have a
Yes. Okay.
no, that's fine. But I
change. Yes, just to change.
Yeah. Okay, but to point.
The pointer is
oh,
here.
okay.
But can use it.
Okay. Sorry. Okay. Okay. So we require our proteins doing metabolism require roughly 15 minerals, roughly 15 vitamins. I won't argue about trace minerals or other things. And then some essential amino acids and a few odd things like choline, and then 2 essential fatty acids, the omega-3s and the omega-6s. And so we think we've figured out the principle of how much we need. And the question is, are we getting enough? And the answer is a clear no. So the committees set up 2 numbers, the EAR and the RDA. The EAR is the estimated average requirement, and that's some distribution in the population. And the committee says if you're below the EAR, you're not getting enough with current standards, which I'll tell you are the wrong standards, but that's another matter. And then 2 standard deviations above the EAR, they set the RDA, which you see on all your potato chip packages, the amount that you require, the recommended dietary allowance. So if you're below the EAR, then there's a danger sign. Well, iron, menstruating women are losing iron, and 16% of them in the United States are below the EAR. Magnesium is a complete disaster. 56% of the whole U.S. population is below the EAR for magnesium. Europe is probably very similar. And where do you get your magnesium? It's in the center of the chlorophyll molecule. So anything green has magnesium. And then if you eat nuts, almonds, say, have both calcium and magnesium. So we're not getting enough of some of these things that are really important, because magnesium is in 500 or so enzymes. Practically every DNA repair enzyme has magnesium. And you can think of lots of enzymes that require magnesium. And then zinc, 12% of the U.S. population. Now, zinc is in 1,000 enzymes with zinc fingers reading particular sequences on DNA. And zinc is in copper zinc SOD. And we're not getting enough according to this. And then of the vitamins, B6, half of elderly women are below the EAR. Folate, it used to be, folate is what got me into this field, and I'll tell you about folate next. But now we fortify the flour with folate in the United States, and still 16% of adult women are low. E, practically everybody. Vitamin C, a third. And then we're very low. They haven't set EARs for these things, though they just did for D and calcium, but I haven't changed my slide yet. But vitamin D is very low. Vitamin K is very low. I'll talk about that. Calcium is very low. Potassium is very low. And omega-3 is very low. So if you just take the 30 vitamins and minerals, which is mainly what I'm going to discuss, it doesn't look right. But nobody cares because there's no pathology. So you can say, why aren't the newspapers having all kinds of stories you're low in magnesium? Because nobody knows what the pathology is. And I'm going to tell you what I think the pathology is. Okay. So this is the experiment that got me into the field. And before I walk you through this, it's a little complicated, I'd like to give the background to it. A fellow named Jim McGregor came to my lab on sabbatical, and he had just done this wonderful experiment. It was published over 20 years ago and should have changed the whole field of nutrition, and everybody ignored it. But it got me into the field with both feet. And what McGregor was doing is he was looking at radiation in mice, and there's a very easy assay to look at broken chromosomes. And everybody knows broken chromosomes lead to cancer. It's the most dangerous aspect of radiation. And what people can do is put red blood cells on a microscope slide and dissolve away the outside of the cell so you're left with the nucleus, and then put a charge across the slide. And so if there's a broken chromosome, the DNA gets pulled out because it's negatively charged, and you can see a little comet. And that's a very simple assay to do, and it's widely used. It measures nicks and breaks in the DNA. Well, McGregor was irradiating mice and looking at the effect of caffeine on DNA repair and things like that. And one day, all his control mice were full of chromosome breaks. And he tracked down what the cause was. Someone had left folic acid out of the vitamin mix. And it was just like radiation. He did dose response curves. Folic acid was exactly like radiation. It was breaking chromosomes. So that he got interested in that. He thought he'd do it in people. And this assay doesn't work in people. It works beautifully in mice, but it doesn't work in people because the human spleen, which is down here somewhere, the human spleen's function is all the blood goes through the spleen, and any cell that's a little stiff gets taken out because the body doesn't want you clogging up some capillary somewhere. And in mice, inbred mice that they've looked at don't have much splenic function, but people do, so you don't see any broken chromosome. This method just doesn't work. Even if you radiate people, it all gets taken out by the spleen. Well, McGregor had the bright idea of going to the local health maintenance organization, Kaiser in our case, and asking, do you have any people without spleens in your database? And you can lose a spleen in an accident, and people do occasionally, and you live perfectly well. So he talked them into giving him the names of 30 people without spleens, and he went to them and found 20 people who were willing to get stuck and take a little blood from them. And McGregor set up the baseline in people. And what he found was he set it up down here. Okay. But then what he found is one guy who had 20 or 30 times the level of broken chromosomes of everybody else. And he said, that's interesting. So he followed him for a year, and he didn't know what was going on. And then when he discovered that folic acid deficiency could break chromosomes, he said, gee, I wonder if this guy is folic acid deficient. And he measured his folate, and it was low, but not very low. It was a level half the poor were in the United States and 10% of the U.S. population. But he measured all these things, and he thought he'd experiment on this guy. So he gave him, these are the levels of folate. He gave him folic acid for a couple of weeks. And the reticulocytes, which are 1% of the red blood cells, are the newly synthesized ones, those you see the changes immediately, and right away it came right down to baseline. And red blood cells, again, which don't have any DNA, but there's occasional piece of a chromosome in, because when the nucleus gets extruded to make, when you extrude the nucleus to make the reticulocyte, if there's a broken chromosome, that little piece of chromosome gets left behind. That's this micronucleus test. And so the red blood cells follow. And then, since he only had given the folic acid for 2 weeks and the guy was eating his normal diet, which wasn't a great diet, the retics started going up and the red blood cells followed. And he went to he thought doing any more experiments on this guy would be unethical. So he said, well, look, you should eat a better diet or I'll give you a folic acid pill. And the fellow said, give me a folic acid pill. And he didn't want to change his diet. And so McGregor gave him a folic acid pill, and both the retics right away and the red blood cells followed came right down to baseline. Now that's a beautiful experiment because you know the level that's doing something. You have established causality. Epidemiology as a field sucks. It's just too difficult. And half the results they get are wrong because they don't understand what's going on and they don't usually measure something. Not to knock epidemiologists, but it's just not something you want to rely on unless you really know what you're doing and analyze it very critically. And it only establishes associations. Where this establishes causality, you see the amount. So for micronutrients, epidemiology is very difficult anyway, but they're good papers and they're good epidemiologists. So I won't slam all the epidemiologists, but it's very expensive and it's a very difficult science. So anyway, this experiment got me all excited because half the poor were at the level of folic acid that were breaking chromosomes. And on one guy, you can establish causality. And you could do it on 5 people if you want, but it really pins it right down, pins the amount down. And this is the way we're going to do RDAs and EARs. Because what you're going to, or RDA, what you're going to do is find out what level of something is doing a biomarker. And in case of cancer, which is a long-term disease, DNA damage we know gives rise to cancer, but it's insidious. And McGregor wanted some help in the biochemistry. He wasn't a biochemist, and one of my graduate students and postdoc worked out the mechanism on why folic acid deficiency breaks chromosomes. There are 2 main pools of folate, the methylene tetrahydrofolate, which was discovered at Berkeley by a friend of mine many years ago, and methyl tetrahydrofolate. And this one methylates DUMP to make DTMP, and so when you're short of this pool, this pool goes up, this pool goes down, and you start putting uracil in DNA. And we set up an assay for measuring uracil in DNA, and you can get hundreds of thousands, if not a million, uracils in DNA when you're short of folic acid. And every time a repair enzyme cruises along the DNA, sees the uracil, it clips it out, and you get a transient nick. And that's the exact same mechanism as radiation. I'll show you in a minute. And then the other pool is methyl tetrahydrofolate, and B12 are converting homocysteine to methionine, and then methionine goes to S-adenosylmethionine, which is the main methylating agent in the cell for making acetylcholine and making methylated lipids and making methylated DNA. And so you're always regenerating homocysteine, and Smith and other people have shown this is a major risk factor for cognitive defect. So high homocysteine has been associated with poor cognition, and it can be reversed again by folate and B12 and B6. And here 3 or 4 types of cancer have been associated with low folate, which fits with DNA damage. And here you have both cognitive dysfunction and heart disease have been associated with this. And then there's a polymorphism. 40% of Northern Europeans have several polymorphisms where they increase this pool at the expense of this pool. Now that's funny because this one, when you're low in this pool, you get cancer. When you're low in this pool, you get heart disease or cognition. Those are long-term things. So why would evolution care? You can folia is a Latin word for leaf. Folic acid was first isolated by my mentor at Caltech from 4 tons of spinach. Foliage is the same root. And so, but you can imagine the people up in Sweden didn't have a lot of big plates of spinach all winter long. But why would nature select for a polymorphism? Well, what about sperm? And we, in fact, published a paper showing that sperm's the thing that nature really cares about. Heart disease and cancer come too late in life. And so what you're doing is you're protecting your sperm by increasing this pool, but the price you pay is cognitive dysfunction and heart disease. Anyway, I think a lot of the polymorphisms are going to turn out to have to do with nutrition. Mm-hmm. Okay. So both Fenwick in Australia and Chantal Courtemanche in my lab compared radiation to folic acid in terms of breaking chromosomes, and this is Fenwick's study. We published them about the same time. And the more radiation you do, the more chromosome breaks, and the less folate you get, the more chromosome breaks. But we're talking about levels that are really significant in the human population, where people worry about incredibly tiny levels of radiation. In fact, they're shutting down nuclear energy in Europe, it looks like, because of a Japanese tsunami. Anyway, whatever it is, if you look at the people who are low folate, it was half the poor in the United States. So this all got me tremendously interested in vitamins and minerals. And so when radiation works because you get clusters of electrons and you get an oxidative lesion on both strands, and then repair enzymes coming along, they see an oxidative lesion, you clip it out, and then you put the right base in, and this strand is holding it together, and you sew it up. But when you have 2 nearby lesions on opposite strands, then you get a double-strand break. And that's what the radiation biologists think. And when you put 100,000 uracils in, and you have 70,000 or so oxidative lesions just from living, you get the same thing. So the next micronutrient we looked at, Fernando Viteri, who's a professor at nutrition at Berkeley, knocked on my door and said, iron. The World Health Organization is giving people too much iron. There are 2 billion women and children in the world who are too low in iron. Up is bad on this graph. I'll get into the details. So we have 2 billion women and children who are getting too little iron, and what that does is foul up your mitochondria. So we were mitochondrial experts, and there was some evidence that too little iron fouls up mitochondria, and Viteri asked us to help them. And so we did the whole range of iron in mice. So too little iron is a disaster. You get mitochondria pouring out oxidants and less efficiency and all these different things. And the red is the sum of all these mitochondrial defects. But too much iron is also bad. So too much iron, in the United States anyway, there are many more women and children with too little iron, and in the world particularly. But too much iron is mainly elderly men who are eating too much red meat. They're getting all that heme iron, which isn't very well regulated. But this is a minor problem compared to too little iron. But too much iron's bad too. And iron is dirt cheap. It's rusty nails. So the World Health Organization is going around giving huge doses of iron to all these women and children who are anemic, and they're giving them too much, according to Viteri. And we kind of showed that too much iron isn't good for you. as well. Now, metals are tricky because they look like metals can be very similar. And so calcium and magnesium, one's above the other on the periodic table. The body really cares about the ratio because you put in a huge dose of magnesium, you can inactivate some calcium enzymes. And you put in a huge dose of calcium, you inactivate some magnesium enzymes. And the body is really monitoring the ratio. And iron and zinc are similar, and iron and copper. So there's a little evidence in the literature, but it's not definitive, that too much iron may be causing you a copper deficiency, or a zinc deficiency. But anyway, that hasn't been worked out yet. But so we should be here, but the world isn't. 2 billion children, women and children is a lot of people. So the next, Emily Ho came to my lab, a postdoc, and she was interested in zinc. So she looked at human cells in culture and made them a little zinc deficient. They pour out the mitochondria pour out oxidants, and in addition, you get all these comets. It does DNA damage. And then we looked at biotin, and we looked at iron. So we looked at a half a dozen vitamins and mineral deficiencies in human cells in culture, and every time we did that, we got DNA damage. I said, what the hell is going on? Why is nature doing that? And I just worried about this for months, and finally I had an epiphany one day what the answer was. It's the trade-off between short-term and long-term. So what we postulated is that built into our metabolism, when you're running out of a vitamin or mineral, even moderate deficiency, what the body isn't, your metabolism isn't static, it's dynamic. So what you do is the proteins that are necessary for survival and reproduction hang on to that micronutrient, and the proteins that are preventing DNA damage shows up as cancer in 10 years. When it's a matter of survival, throw it overboard. It's not important when it's survival. And I think all our metabolism is designed so when you get moderate deficiencies of vitamins and minerals, the price you pay is long-term damage, and what you get out of it is survival and reproduction. That's the idea. And magnesium deficiency gives you mitochondrial DNA protein cross-links. And so if you just put into Google the 30 vitamins and minerals and mutation, cancer, DNA damage, a whole world of studies comes up. And again, it's epidemiology and human cells and culture. Various people have done various things, but there's evidence for all these vitamins and minerals that you get DNA damage. And I said, gee, does everything get DNA damage? And what's doing this? And then I called this idea triage. So, and we published a paper about 2006, and I can't say it got the nutrition community all up and cheering. It was pretty much ignored. But anyway, so we decided we're going to prove it, and we're proving it from lots of directions, and that's what I'll finish up telling you about, that I think it's really going to be true. So I have this wonderful scholar in my lab, an older lady who used to be a postdoc with me about 35 years ago, and she came back in the Bay Area and was looking for a job, and she's really smart. And she can do all sorts of things I couldn't do in 100 years. Anyway, she can analyze 500 papers and know whether the mouse studies are any good, the epidemiology is any good, and does it fit with the metabolism, and write a definitive review on it. Joyce McCann's her name. And so she came to me one day and said, I'm a little skeptical of your triage idea. I think there's a better way of tackling it. We'll see if it's really true. So I said, Joyce, go to it. And so what she did, she likes doing theoretical work. So she pulled out those vitamins and minerals that aren't too complicated. Vitamin D goes to a hormone. If you don't get your sunshine, you're deficient in vitamin D. Ultraviolet light in your skin makes 25-hydroxyvitamin D, which makes a hormone calcitriol. And there are 900 genes that have a sequence in the front of them that need calcitriol to turn them on. So you don't get your sunshine, or you have a dark skin in a northern climate, you're in deep trouble. And but so you don't want to work on that. You'd spend the rest of your life in the library trying to figure it all out. So instead, what she did is pick ones that weren't too complicated. And she's completed vitamin K. We published that 2 years ago. Selenium's just coming out right now. And the only way to understand these is from this triage perspective, that there's moderate deficiency trashes long-term things as the price of surviving. So I'll just go through vitamin K briefly, and then selenium briefly, and then pack up. Okay. So how many minutes do I have left? 5. Okay. So vitamin K, K stands for coagulation in German, as many of you know. It was first discovered as a blood clotting factor. And what it is a compound that's used in photosynthesis in plants, and phylloquinone. And animals have hijacked it for a different purpose. It's a cofactor for an enzyme that takes an already made protein, and when there's a glutamic in the right context, it puts an extra carboxyl group on the glutamic. So you need this cofactor, phylloquinone, for doing that. And that amino acid is called GLA, so you're changing a glue to a GLA. And if a protein has a GLA in it, you know vitamin K was involved. So all the coagulation proteins have GLA in it. But there are a batch of other proteins that don't have GLA in it. So what Joyce's wanted to do is see which were the essential of the 16 vitamin K dependent proteins, which ones were essential and which ones were non-essential. So she went to the mouse knockouts. And by now, they've knocked out most of the genes in a mouse, and somebody's published a paper, what happens? Well, you knock out the coagulation protein. So you knock out the coagulation proteins, they're all embryonic lethals. Those are essential. And you look in the human gene, you see that fits too. And that makes sense. You cut yourself, which you do fairly often, and you'd bleed to death if you didn't have all these coagulation proteins. And calcium's involved in the blood coagulation. That's why they're all calcium-binding proteins. So those are essential. So that's a pretty crude measure of essentiality and non-essentiality, but she used it for this. But then so we don't care about those. The interesting ones are these. And the hypothesis is that you need more vitamin K to do these proteins to do these coagulation proteins than you do these long-term things. And that turns out to be true. So we didn't do any of this work. It was done by terrific people who spent their lives on vitamin K, particularly Sugars and Vermeer and Maastricht in Holland are doing wonderful work, and Sarah Booth in Boston. There are lots of really good people in the field. We're just looking at it from a different perspective. So you knock out each of these genes, what happens? Well, the animals are born and they go on their merry way. Except that you take the ones with the osteocalcin gene knocked out, and you stress them a bit and their bones break. Osteocalcin's in the bone, and it's moving calcium around in the bone, and you can make bones without osteocalcin, but they're not strong bones and they fracture. And so vitamin K deficiency is associated with bone fractures. So that fits. And you look in the human gene, there's polymorphisms that kind of fit. So GAS6 is acute coronary syndrome. Matrix GLA protein is a calcification of the arteries. TGFBI, they all get cancer. So it's all long-term things. So I'll just go through these briefly. So you can look at osteocalcin, function, bone structure, mouse knockout phenotypes, fragile bones, human mutants, SNPs have bone fractures, anticoagulate. There are 30 million prescriptions written every year in the U.S. for warfarin, or it's also called Coumadin. And what that does is interfere with vitamin K metabolism because people are dying of thromboses, so this kind of thins your blood. It makes you less likely to get a thrombosis. But the price you're paying is you're trashing all these other enzymes. So, so the people who are getting Coumadin or warfarin have more bone fractures. Men, children, rats. Minus vitamin K deficiency gives you bone fractures. Now, the really interesting one is matrix Gras protein. So there, the animals are all born and they look okay, and then they all die of calcification of the arteries. So it turns out the function of this protein, whenever it sees calcium coming out in the kidney or the aorta or the blood vessels, is it grabs it and takes it out of there. And and calcification of the arteries is a major risk factor for heart disease. So, and you look in the human gene, there's a rare genetic disease called Coital syndrome where people die at an early age of calcification of the arteries. So again, everything fits. In addition to all of this, and the people who get Coumadin die at 9 times the rate of calcification of the arteries. Okay, and the docs don't know this. They'll give you Coumadin, but they don't know about all this stuff. So it's really has all these medical implications. Anyway, and then one of the other genes, this gene at the front of it has a little sequence to turn it on. And what turns it on? Vitamin D hormone. And one of the things that they know about vitamin D deficiency associated with calcification of the arteries. So there are 2 micronutrients involved. This is natto, which is a Japanese health food. It's a B. subtilis fermented soybean. Now, the way the body works it is when all the coagulation proteins are done in the liver. These other proteins are done in the non-hepatic tissue. When you eat greens, which is where you're getting your vitamin K, it goes right to the liver and does the coagulation protein. And then the phylloquinone is converted to a menaquinone, very closely related molecule. And menaquinone has the property of going to all the tissues in the body equally. So the MK series goes to everything equally, and you do these other proteins, but you need a lot more vitamin K to do those. So, and natto is filled with MK7. It's a B. subtilis fermented soybean, and they've done the epi in Japan. The people who eat natto get very little heart disease. They don't get bone fractures. Everything kind of fits. And they did a study, and one of the other genes, TGFBI, you knock that out, all the animals die of cancer. And we understand the mechanism because there's a lot of aneuploidy, and that protein's in the matrix interacting with integrin on the surface, which interacts with the microtubules, so your mitosis isn't as accurate. So right away out of all this, you understand mechanism. Now, if you want to do epidemiology, you can measure carboxylate versus uncarboxylate, and the Maastricht group is doing this. And so, and you need more vitamin K to do these long-term things. So it's all long-term, and it's sort of understandable. Now, I'll just say a word or two about selenium. Again, selenium is all long-term. There are 26 proteins that involve selenium. Lots of good people have spent their lives on this. But you do look at the knockouts or all of that. Joyce McCann did all this. It's all coming out. But it's all long-term stuff, selenium deficiency, heart disease, DNA damage, cancer, cognitive function. And again, you understand mechanism right away. So I think we're going to have to work. It's also complicated. We're going to have to work from mechanism on up, but now we'll know what to do. And every micronutrient we've looked at uses different mechanism to do the partitioning and do the triage of which gets it and which doesn't when you get moderately deficient. The way selenium works is that it's a modified transRNA that has selenocysteine in it, and there's a modification on the tRNA. Many years ago, we worked on tRNA modifications as a regulatory circuit. I think it's a very early system of regulation, but that's another matter. And so there's a methyl group here, and the tRNA that has the methyl group will do these long-term things, but the essential ones can be done by the tRNA without the methyl group, and selenium deficiency you can't put on the methyl group. So it all, right away you understand the mechanism, you understand that there are two groups of genes, the essential ones and the non-essential ones, and it's the non-essential ones are all these long-term things. So the conclusion from all of this is this provides a rationale for why modest micronutrient deficiencies might increase risk of disease of aging. So we're looking in the wrong place. We're calling vitamins and we're only fixed on early death. That's how we discovered all the vitamins, beriberi and scurvy. But now we want to live a long lifespan, and it's all these long-term things that we're cared about, and we have to optimize micronutrients for that. And so it offers a strategy for setting intake recommendations and for developing relevant biomarkers assays. So the future of disease prevention, or a lot of the degenerative disease we're associated with aging, are going to be, there'll be a little machine in every pharmacy in the world. You put your finger in, it'll take a finger prick of blood, and the analytical methods are getting so good now, you can measure any protein in human blood at great sensitivity on a finger prick of blood. There are 3 or 4 companies that each one has some fantastic technology. And Dean Jones here was telling me he can measure 4,000 human metabolites on a finger prick of blood. So all these methods, so you're going to put a finger prick of blood into this machine, and it'll send the results to your iPad, and you cut out expensive doctors, you cut out expensive phlebotomists, and we'll do preventive. It'll tell you, eat your spinach or take a pill. And now whether you can get people to do that is another matter, but at least we'll understand how to do it. Vitamins are cheap. On the other hand, don't overdo it. You take too much selenium, you're going to poison yourself. Mae West, the American actress, said, too much of a good thing is wonderful. But she was thinking about sex, not micronutrients. So you don't want to overdo it, particularly in the minerals. And the last, it suggests where to look for a mechanistic connection between micronutrient deficiency and disease. Just a word about the immune system before I pack up. Okay. So we entered into Google immune system and what goes wrong with aging. And these 6 things popped up. Okay? And then I entered each of these against the 30 vitamins and minerals and asked what I got. And what I got was this. So there's literature out there, and it's your adaptive immunity goes out when you're short of vitamins and minerals. And in some ways that was known, but they didn't know quite the mechanism. But it makes sense because so you don't remember the virus that hit you 4 years ago. But when it's a matter of survival and reproduction, who cares? That's basically, so nature's always, you get a little low on something, you don't eat your green, what do you get from your greens? You get your folic acid and your vitamin K and your magnesium, and Americans aren't eating their greens. And so.
We have about 30 more seconds.
Okay, good. So I'm getting to the end. So this is one of Fenwick's studies. Fenwick's also been looking at DNA damage and chromosome breaks and things like that. And these are healthy, non-smoking men and women. Women live longer than men and have less DNA damage. But look at that variability. That's what we ought to be going after. And I think most of that's going to be due to nutrition. Nutrition's the big thing. The low-hanging fruit is in disease prevention is in nutrition. And that's what we have to eat better diets, more. The whole purpose of a balanced diet is to get all these 30 vitamins and minerals and the other things you need. And we're not doing that. We're eating too much refined food. One sugary soft drink is a disaster. We said that's the main source of calories in the United States. It fills you up and you don't get your vitamins and minerals. Plus you get a leaky gut and bacteria goes through and gets you all inflamed and you get fat. And plus, I think it makes you hungry when you're short of vitamins and minerals. Anyway. Okay. So these are the people in my lab. Jung Soo, who's a wonderful postdoc who's just becoming independent, Joyce McCann, who's this scholar who's done all these reviews, Swapna is an Indian postdoc working on the immunology. David can do all the minerals at once by ICP mass spec, and we can study that. There's an MD who helps on some things. Sandy's forensic biologist interested in mitochondrial DNA, and we're collaborating. Mark is my Buddha. He's a Japanese-American with a big round face and a big round belly, and he's the wise man in the group. And I called him my Buddha. But his blood was the worst. He had low HDL and high LDL, and it was a big mess. So he's exercising now. So he doesn't look like Buddha anymore. And Michelle and Ash are two physicians who help on our clinical trials. We've made a bar with all sorts of goodies and fish oil and magnesium, calcium, and enough. A vitamin pill is a good first multivitamin mineral. It's a good first try, but it doesn't have enough magnesium, calcium, potassium to make the pill too big, and it doesn't have fish oil. So you have to really think about all these things. And anyway, thank you.